as we look at cell membranes, the place to start is with membrane structure. And for membrane structure, we have a number of molecules that make up the structure of the membrane itself. Now, the membrane is often referred to as a phospholipid bilayer, but that's really only part of it. It is the core structure of the membrane, and so that's where we really need to start, is with phospholipids, the behavior of phospholipids, and how the phospholipids form what is the core structure of a cell membrane. Once you understand that and the concepts related to it, then we can add in all the other pieces. Once we have the other pieces added in, then we can see their job and their role in the membrane itself and how the membrane functions for a cell. And the membrane is an incredibly important part, obviously, of a cell. There is no cell without the membrane. All living things are cells or are made up of cells, and the cells are bordered by and created by membranes, and then the membranes make up all the substructures inside the eukaryotic cells as well. Actions and activities that take place within cells often happen within compartments or between compartments within the cells that are made up of membranes. And then the action often of proteins embedded in the cell membrane or attached to the cell membrane, or what carry signals between cells, allow cells to attach to other cells, allow the cells to change shape and behave, and a variety of other things that we can only get at once we understand the structure of the membrane. So we can't get into any of the function or activities until we get its structure. So phospholipids, we already should be familiar with this, right? But we'll go over really quickly what is a phospholipid. So the phospholipid structure, again, a little more detailed structure. We have a 3-carbon molecule called a glycerol. The glycerol has OH groups attached to it, like this. The glycerol then has fatty acids attached to it. There are 2 fatty acids that get attached down here. Okay, I'm going to take one of these away. We get another one here. Now, how many carbons long? These are variable. Okay, so this can be, 13 carbons, 15 carbons, 16 carbons, different numbers of carbons in length. The carbons themselves may have double bonds between them, and they may not. But in general, all the carbons are going to have hydrogens, and if they don't have hydrogens attached to them, that's because there are double bonds there. So instead of having 2 hydrogens attached like this, there's only 1, 2, 3, 4. So there's only 1 hydrogen attached there to each of these carbons. So they're not saturated, so we call those the unsaturated ones. So this should be review. I'm not drawing in all the other hydrogens for all these. It just takes really too long, but you should already know this from previous lectures, right, on the structure of a fatty acid, what a fatty acid is, and the overall structure of a phospholipid. So we have a glycerol, 2 fatty acids, and then up here is the phosphate functional group, which we see this now all over the place, right? Phosphates are part of the DNA. Phosphates are part of ATP. Phosphates are part of the phospholipid. So make sure you understand a phosphate, what it actually is. And then attached to that phosphate is some polar molecule, some sugar, alcohol type molecule, a variety of different things. That's usually unique. The name for the phospholipid usually comes from that polar molecule, like phosphatidylcholine. So this polar molecule might be choline, and that's kind of the name where the name comes from. We're going to depict the phospholipid in a little more simplistic way. So this whole region up here, that's polar. These are charges. That's polar. This is polar. These are polar bonds. This is a polar bond. We're going to call this whole region a polar region. Now these 2 tails sticking off here, it's carbons, carbon and carbon. The bonds are sharing electrons equally. They're non-polar. So they don't create any charge. The bonds between a hydrogen and a carbon, they're equal or are almost equal. So they share equally with each other. They don't create charge or partial charge. So this whole region is nonpolar. So when we draw a phospholipid, we often draw the polar head like this, and then we get 2 squiggly nonpolar tails. Right. That's a phospholipid. All right, that's how we'll often now, as we start to draw bigger structures of membranes, too much to draw the overall structure, we need to simplify it into something like this. But this is what we're actually talking about. You have to be familiar with it to know what it is. Phospholipids, in general, are oily substances. So if we were to take a phospholipid, like I mentioned, phosphatidylcholine, and have it in a container, it would look something like, say, olive oil. And then we pour it into, say, water. And what would happen is, if we had a container, see it like this. Okay, and in the container we had water. And then we poured the phosphatidylcholine into the water. What's going to happen is that the tails of the phospholipids are going to go down. Sorry, flipped it around. The tails are going to hate the water, and they're going to stick up into the air. Okay, so that one is all messed up. So here's our water. Let's do this again. So it's a good practice to kind of think about this. All right, this is water in here. If we were to then add phospholipids to the water, remember the tails, they would hate the water. They don't like the water at all. So the polar heads, which have charge and or are polar, they will interact with water molecules. But the tails, which have no charge, don't like water, and they're going to actually stick up into the air like this. Okay, so they're going to stick up out of the water. So they would form almost like an oily film. What you would see as an oily film, say, over the surface of the water, like as if you would pour olive oil and water together, you see that oil separation. Now, the unique thing here is that with the something like a triglyceride, like olive oil, it's pretty much all nonpolar. But here we do have a polar part. So if we were to take this and then we were to cover it, all right, and we were to shake it up and mix it and start to mix these molecules around, something would happen, all right, to the phospholipids. They would change, all right, their orientation. They wouldn't all just stay as a little layer. What would happen is that many of the phospholipids would join together into little structures like this. So each of these is an individual phospholipid, polar head, two nonpolar tails. And we would get something forming like this. This is a structure called a micelle. Now this would be a three-dimensional sphere, okay, not just a little flat structure like this. And as a sphere, its outer surface, like a ball, would be covered with the polar heads of the phospholipids, and the inside, the interior of the ball, would be all nonpolar tails. So this whole region in the interior is nonpolar. It's a nonpolar interior. And that's called a micelle. Micelles can form naturally in the ocean. In nature, we see them. We could do it in a laboratory. Micelles will just naturally form because of the association of the polar heads in water and the nonpolar tails to each other. Now, there are no bonds here between the tails. It's called nonpolar association. They just don't like water. Water doesn't like them, so they come together and they aggregate, and that forms somewhat of a stable structure. Something else will happen is that these, shaking it up, we can get some larger structures to form. We could also get something like this, where looks sort of like the micelle, where we have the phospholipids, polar heads, nonpolar tails, sticking inward like this. But then we see a second layer. All right, another layer of phospholipids. But this layer is a little bit different. The tails are going to orient with the other tails. Like that. So now this whole region through here, this whole entire region, is nonpolar. But we have something different. At the core, right here, the middle of this structure, it's surrounded by the polar heads, which means water can be in here. It has a polar interior. This is a structure called a liposome. Liposomes can also just form spontaneously. In nature, you can mix up phospholipids and form liposomes. What the liposome just formed, though, as you can see here, is phospholipids oriented opposite one another. The tails orienting like this into what we call a bilayer. So bi meaning two layers of phospholipids. But the two layers of phospholipids are not just equally stacked on top of one another. One is flipped so that the tails are joining the other tails. They don't actually physically bind. There's no chemical bonds. There's no hydrogen bonds. There's no ionic bond. There's no actual bonds. It's non-polar associations between the tails. There's hydrogen bonding between the heads and each other. There's hydrogen bonding between the heads and water molecules around them. So there can be water over here. There could be water over here, but there's no water here. This layer excludes water. And that's the beginning of starting to talk about cell membranes, is that the phospholipids themselves can be oriented into bilayer structures. So we begin with what we often refer to as the phospholipid bilayer. A phospholipid bilayer is not a cell membrane. A cell membrane has all these parts to it. Okay, so for a living cell, it does have a phospholipid bilayer, but then attached to it and integrated within it are all these other molecules too. But this is the place to start. Okay, so we have micelle is a single layer of phospholipids. A liposome is a double layer, forming a little ball or sphere, but it's not a true membrane because we don't have any of the other things attached to it yet. So where do these other things go? We can kind of continue with this a little bit here. So if we expand our phospholipid bilayer a bit, make it a little bit larger, and then we start to study it. We'll find a couple things. One, relating back here to the fatty acid tails. These tails here could be saturated or unsaturated. And there could be all sorts of combinations. So the tails here within the membrane, this one could be saturated, this one's unsaturated. Most of them could be unsaturated. Most of them could be saturated. That is going to vary between different organisms, between different cells. The composition of them varies for a number of reasons. Some of them are adaptations of the cells to the environment in which they live in. Helps the cell become more stable in a particular environment by having more or less. Helps the fluidity of the cell, which is a concept we're going to get into in a little bit, about how flexible the cell membrane is, if it has bent, unsaturated tails or straight tails. That will allow the phospholipids to be packed together in different ways, or they're tight or they're spread apart. So that's a concept of fluidity, which we're going to get into in a bit. So we're still focused on just structure though right now. We have phospholipids, a bilayer. Next we have cholesterol. So cholesterol molecule, I'm not going to go into the full detail of the cholesterol molecule, but we're just going to give you the overall structure. Looks something like this, okay? So a cholesterol molecule has this ring structure. It's called a cyclopentophenanthrene ring structure. So again, these are all going to be carbons. All carbons, it's all carbons. Carbons and hydrogens, okay? The whole thing. So non-polar. And then it has an OH group here. Cholesterol molecules are then going to be found in between the tails, obviously, because they don't like the heads. They're all really non-polar. They're about as big as some of the tails, depending on the particular size, the length of the number of carbons, and the length of the tails. But we typically find cholesterol molecules here between the tails. And you could find cholesterol molecules on both sides of a bilayer, like this. The concentration of cholesterol molecules can also differ. You could have a lot of cholesterol, you could have very little cholesterol. But one of the roles of cholesterol is to be part of a cell membrane. So you've heard of cholesterol. Most people think of cholesterol in terms of diet. Most people think of cholesterol that's free in the blood that can then cause clots within blood, and thinking of it as a negative thing. But cholesterol, this molecule itself, is a precursor to many different types of hormones. and cholesterol is an essential part of the membrane. So we'll get into a second here. Why is cholesterol essential part of the membrane? And it's related to that topic I brought up called fluidity. So for a minute now that we have cholesterol and the phospholipid bilayer, we're going to stop. We're not going to get into the glycolipids and proteins yet. We're going to talk about this concept of fluidity first. So a membrane is not a hard, solid barrier. You have to think of it as like an oily layer, right? So the phospholipids like an oil covering the surface of water, in this case forming, say, a ball or a sphere that forms a cell membrane that contains water on the inside and then has water on the outside. That little oily layer, that's the membrane, right, then can either be somewhat tight and rigid, or it could be very loose and flexible. Right? So it's a concept that we call fluidity. So fluidity is really how flexible the membrane is. Things that affect fluidity. There's going to be environmental things that affect fluidity, like heat. The warmer anything is, the more molecules move. The faster they move and the more they can move from each other. So if you increase heat, you typically increase the fluidity. The membrane becomes more flexible. If you decrease the heat, the molecules move slower and tend to be closer together, and the membrane becomes less flexible. It becomes more solid and rigid. Okay. So heat of the environment is going to change it. Some organisms live in a constant temperature. If you live in cold water in the ocean, that may not change at all, ever. It's very static. Other environments, other organisms live in temperatures that fluctuate every single day in different seasons of the year. So they may have to adapt to this by having different amounts of cholesterol or different changes to the fatty acid tails, so which we'll get into in a second. So heat affects fluidity. And then other things that affect fluidity are going to be the fatty acid tails. Again, I'll just do it like this. Here. Tails that are unsaturated are bent, right? And they push far away. Tails that are saturated means that they're straight. So straight tails, so think of pencils. So you could take a bunch of pencils and put them really close together and pack, just like these pens here. I could put them really close together. But if they were bent and wavy in all different shapes, I may not be able to fit them all tightly together like this. There's going to be gaps between them. There's going to be spaces that push them apart. So unsaturated ones, because they're pushed more apart, makes it more flexible or more fluid. Saturated ones make it less fluid. So the fatty acid tails themselves can affect the fluidity. Organisms that often live in warm environments have more saturated fatty acid tails because the heat of the environment is starting to make the membranes more and more flexible. But the cell has to then counteract that so it doesn't become too flexible and just fall apart. So they tend to then have straighter tails. Organisms that live in colder environments, the cell membrane then tends to become less flexible because of the cold, right? More rigid and solid. But the cell has to be able to move and change shape. So then those cells will counter it by having more unsaturated tails so that they maintain some of that flexibility. So we kind of have fatty acid tails as part of it. And then the third thing really affecting the fluidity is going to be the cholesterol. So how's cholesterol going to affect this? Well, you could think about it like this. The cholesterol, if it's inserted here, I'm kind of reversing my colors here, but you can see if I put the cholesterol now in between the fatty acid tails, it's going to kind of push them apart. So now maybe in this drawing here, I'm going to spread apart some of these. I'll spread them apart a little more here by putting cholesterol molecules in between some of them. So where there's no cholesterol, they're tightly packed together, but where there is cholesterol, they get pushed apart a little bit. So cholesterol could potentially then increase the fluidity of a membrane by providing little gaps and spaces in between the phospholipids, allowing them some space to move. Right? And so at colder temperatures, where the membrane becomes really tightly packed, another way of keeping the membrane flexible is by having more cholesterol, so that the cholesterol can kind of allow this pivot and movement of the phospholipids a little bit. So cholesterol can make it more fluid at low temperatures. Now, as temperatures increase, the phospholipids are going to want to spread out, right? And then they're going to start to become so spread out that the membrane itself could fall apart, and then the cell will die because the membrane becomes ruptured. Cholesterol can, and this is the harder part of this, the concept to get. Cholesterol molecules, if we put this in a more of a three-dimensional shape, which I can't really draw up here, can also sort of create groupings. Okay, so it's like these phospholipids here, these three in between those two cholesterols, are in a group. So now as the temperature increases and everything starts to pull apart, these three, though, are kind of kept closer together because of the cholesterol, right? Because of the different cholesterol molecules and within the membrane staying in place, not allowing that movement. So cholesterol can counter then heat in a way by making them less flexible, less fluid at higher temperatures. Because of this, we talk about cholesterol as being what we call a fluidity buffer. So buffer, this is the key of this word. So what's a buffer? A buffer is something that helps, say, chemical molecules, a solution, sort of maintain its properties even when the environment is changing. So for example, a pH buffer. There was pH earlier in this course. You should have studied. pH is a change in hydrogen ion concentration in a solution. A pH buffer is a chemical that's also present in the solution. And as the pH changes, it counters it. So essentially, if hydrogen ions are added, the buffer removes them. It will bind to the hydrogen ions and take them away. So something's adding hydrogen ions, but the buffer who's present is taking them up, so the pH doesn't change. Even though an acid is being added, the pH doesn't change because the buffer is preventing it. It can happen the opposite way too. You could try to add a base to it and then it would become more basic, but then the buffer might add hydrogen ions to it counteracts whichever direction something is changing. So in this case, heat is the driving factor. Heat is starting to influence the membrane. It's starting to pull things apart because it's hotter, or it's trying to crush things together or slow them down because it is colder, right, and becoming less flexible. But the cholesterol is that buffer in between. At the cold temperatures, as things get packed, it stays between them, allowing some flexibility. As things heat up and try to expand, they get stopped or blocked by the cholesterol, so they can't get as flexible as they would be. It's a buffer, right? It kind of counteracts a little bit what temperature does. Really high temperature doesn't matter with the cholesterol. You can destroy a membrane. Really cold temperature, doesn't matter if cholesterol is there. You can still freeze a membrane and solidify it. So it's not going to completely stop the action of temperature, but it can buffer it to a certain degree to make it a little more flexible in the cold and a little less flexible in the heat, in the high heat. Right? And so cells that then live in, or organisms that have cells that live in extreme environments will have different amounts of cholesterol to help buffer the temperature, right? Temperature's effect on membrane fluidity. So sum it up. Phospholipid bilayer. Phospholipids oriented opposite one another with the tails toward each other. No bonding, but we call non-polar association between them. Water's on the inside, water's on the outside. They can move. They can kind of float around. Right? Typically, you don't flip-flop. You don't have this phospholipid go from one side to the other, but they can kind of move back and forth, and that's what we call fluidity. How fluid are they? Depends on a number of things. The tails themselves, how bent they are or how straight they are. Unsaturated makes them more flexible. Saturated makes them less. Then the cholesterol. Adding the cholesterol in between the tails can push them apart a little bit. So in the cold, it makes them a little more flexible, a little more fluid, right? But in the heat, when they would normally be pulled apart, the cholesterol can actually counteract that in a way that keeps the phospholipids together. So we call cholesterol a fluidity buffer. So that's the first part. So the first part of this is putting together the phospholipids to form a bilayer and looking at this characteristic of fluidity and things that affect it. The next thing we're going to get into is we'll start to add in the other molecules, and we'll start to talk about one of the primary jobs of a membrane, which is acting as a barrier between one side of the membrane and the other, allowing things to be trapped maybe in one place and allowing the environment in the cell to be different from the environment outside the cell.